Now that we have discussed DNA, let's look at how it's packaged into chromosomes. As always, here are the objectives for this topic. I expect you to have these mastered by our next exam. In this topic, we're going to look at chromosomal structure and regulation. We'll be covering the subtopics that are listed on this slide. Throughout this lecture, I have mentioned that the entire genome of an organism is packaged into every somatic cell. The question then arises, how do cells manage to keep this much genetic information packed into one cell? Just how much physical material is in one human cell? Well, the human genome of one individual, if unwound and stacked end to end, would extend approximately 2,000 miles. And each cell in your body contains that much DNA. Given its length, it is important that DNA packaging is done in a precise manner. The cell must not only package DNA tightly so that the chromosomes may be easily moved around during cell division without becoming tangled, they must also ensure that DNA in the cell can be easily accessed so the code can be read for protein production. The answer to this tricky balance is the packaging of DNA into structures known as chromosomes. As covered previously, chromosomes were identified by scientists before DNA had ever been identified. As far as we're aware, chromosomes come in two different structures, linear and circular. Prokaryotic chromosomes tend to be circular, while eukaryotic chromosomes tend to be linear. Let's take a look at the human chromosomes. Different organisms have different numbers of chromosomes. Just because an organism is complex and highly evolved does not mean that it has the most chromosomes. For example, we humans have 22 pairs of chromosomes with 2 sex chromosomes. The king crab has 208 chromosomes. Back to humans. It is important to be able to identify each pair of chromosomes for identification of genetic diseases. This process is known as karyotyping and is performed by a cytogeneticist. Mostly, these assays are performed on neonatal samples to ensure the health of the fetus. Karyotyping will identify diseases ranging from too many or too little chromosomes to large-scale translocations, deletions, or insertions. In order to perform a karyotype, the chromosomes must be isolated in early mitosis, when the chromosomes are tightly condensed and easily identified. Besides ensuring that only 22 pairs of chromosomes, plus the sex chromosomes are present, a cytogeneticist also looks at the length of the chromosomes, the position of the centromeres, and the banding patterns. There are 2 techniques utilized in karyotyping. The first of these assays is through the coloring of chromosomes via DNA hybridization. Similar to the microarray assay we talked about earlier in this chapter, sequences of DNA derived from each chromosome are tagged with a color and placed in the presence of the chromosomes. This results in the entire chromosome being stained a specific color. The chromosomes can then be sorted by color and length to identify each chromosome. The key to this assay is the specific sequences of DNA that have been derived from each chromosome. If the sequences were not chromosome specific, we would have mixed staining and would not be able to identify each chromosome easily. It's important to note when looking at a karyotype such as this, the chromosomes are artificially lined up. We do not isolate chromosomes from the cell in lined up patterns such as this. An experienced cytogeneticist can do this process relatively quickly in the laboratory. One of the bonuses to this process is that you can quickly tell if there are too many chromosomes or not enough chromosomes. There is a new genetic test available prenatally that takes the blood sample from the mother and can identify and ensure that there are the correct number of chromosomes for the mother and for the baby, allowing us to determine the presence of monosomy or trisomy early in pregnancy without the invasiveness of amniocentesis. The older way of doing a karyotype is through a banding dye. In this case, the dyes are specific for regions that are heavy and either adenine or thymine or cytosine and guanine. These dyes will bind to those segments on the chromosome. This creates a unique banding pattern for each chromosome that allows the chromosomes to be sorted quickly. It is important to note that these bands do not correlate to genes. And while there may be differences in genetic code from individual to individual, the banding patterns are surprisingly similar. One of the bonuses to this process is that we can identify if main bands are missing or have been translocated. Neither this banding dye nor the fluorescent dye used in DNA hybridization can identify anything more than macroscale genetic issues. For specific genetic diseases, further testing must be performed. But these forms of karyotyping allow us a way to easily screen individuals for potential problems. I briefly touched on this concept a few times in lecture so far, but it's important, so let's spend a little more time on the organization of DNA in chromosomes. All cells in the human body, and for most organisms, there are sets known as diploid. That means that they have 2 copies of every chromosome. Because humans are diploid, we have 22 pairs of chromosomes plus 2 sex chromosomes, which adds up to 46 individual chromosomes, 2 of each type. These pairs of chromosomes are known as homologous pairs, meaning they code for the same gene and are nearly identical and have the same genes on them in the same order. The genetic variation we see within the gene does not change the fact that the chromosomes are homologous. Remember, homologous refers to the carrying of genes, not necessarily what the variation of the gene is. Notable exceptions in humans are the egg and sperm cells, which are haploid, which means they carry 1 of each chromosome instead of 2 of each chromosome. They have 23 chromosomes rather than 46. It's important that eggs and sperm are haploid because they will join together to form a zygote, and if they are carrying diploid numbers of chromosomes, the offspring would have twice as many chromosomes as needed. Thus, it is very important that sex cells undergo the process of meiosis and use this to divide the chromosome count in half. Red blood cells take this one step further and contain no DNA because they spit out their nucleus during their development. Also, some organisms are known as triploid, meaning that instead of pairs of chromosomes, they have sets of 3 for each chromosome. Now let's discuss how chromosomes and genes are related. Chromosomes contain genes in a linear fashion, but they are not spaced evenly, or even one right after another. Instead, there are regions of DNA in between each gene that have been named junk DNA. This naming arose when researchers first discovered these structures, but couldn't determine their function. In recent experiments, researchers have discovered that these segments of DNA help control regulatory genes, as well as playing a role in other functions related to the reading and replicating of the genes. Before we go much further, we should define a gene. A gene is a given stretch of DNA that codes for a polypeptide or an RNA sequence that has a specific function within the organism. As we saw in the human genome slide, the human genome contains approximately 25,000 genes. While there is somewhat of a correlation between organism complexity and the number of genes, this is not always straightforward. One reason for this is the nature of so-called junk DNA. These segments are highly conserved and seen in most eukaryotic organisms and serve as a spacer between the genes. An example of how complexity and the number of genes are not necessarily related can be observed in a number of plants and amoebas. These single-celled eukaryotes and plants contain many more genes than we do. So remember, just because organisms are highly complex does not mean that they have a large number of genes. This slight correlation also plays out in chromosomal numbers. Often organisms will group their genes into any number of chromosomes. One great example of this is highlighted in this picture. Here we see 2 different deer. The Chinese version has a genome that is condensed into several small chromosomes. In comparison, the Indian species has the same amount of DNA but has the DNA condensed into fewer and larger chromosomes. Remember, both deer have the same amount of DNA but is packaged differently. The genes of a given organism and its chromosomes have been shaped over millennia through random genetic events and selective pressures. As you can see in the human karyotype, all of our chromosomes have significantly different lengths, and these lengths have been selected for over the course of our evolution. Chromosomes exist in different states within the cell. This is because the cells are in an ever-transitioning cycle known as the cell cycle. The only cells that don't continually cycle are those in our body known as quiescent cells. These cells stay in the G0 phase. We will discuss that in more detail in Chapter 18. I'll post a Blackboard video about the cell cycle in case you can't wait until then and would like a refresher on the cell cycle. At any given time, a majority of the somatic cells in our body are in a phase known as interphase. This is a phase where the cell will prepare for cell division. While there's a lot of preparation for cell division, the main focus and concern for us is the DNA replication phase known as the S phase. Cell division takes place in the mitotic phase, or in the M phase for short, and we will briefly discuss that as well with regards to DNA replication, but we'll cover the entire process of mitosis later this semester. In interphase, chromosomes exist as long, loose threads that cannot be distinguished from one another using a standard light microscope. However, while these threads are loose, they occupy certain areas within the nucleus. As you can see in this picture, we have stained the chromosomes using the karyotyping stains we talked about earlier. As you can see, the different colored regions indicate where each chromosome can be found. Also within the nucleus is a region known as the nucleolus. In this place, there is densely packed DNA known as heterochromatin, and we will discuss that in a bit when we talk about DNA folding. Well, this section is running a bit long, so I'll be breaking this topic into two parts. Take a break and come back for 2.1.